Imagine being able to create enough blood for a human transfusion from a small patch of your own skin. That's the idea behind a new procedure that's being developed by Canadian scientists. Now, they were able to take a mature human skin cell and turn it directly into a blood cell without having to rewind the cell back into a stem cell. That means that there's no chance of rejection in transplanted cells because they're your own. Now, for years, their research focused on creating stem cells, but when that proved to be inefficient at creating cells for clinical use, their focus went to creating direct conversion. Basically, what you're doing here is reprogramming a cell. That means patients who need blood, and that could include bone marrow, could have access to their own blood. Dr. Mick Bhatia is the director and senior scientist at McMaster University in Hamilton, Ontario. He's the study leader. Dr. Bhatia, thanks very much for joining us. That was our attempt at making sense of something that you and your team have been working on for a long time. How accurate is that?
It was spot on. Perfect.
This sounds remarkable. It sounds really groundbreaking. Tell me about what of this has been able to has been done before and where you've taken this advance.
Well, it's been shown before that a Japanese scientist by the name of Shinya Yamanaka was able to take skin cells and turn them into stem cells, but those stem cells then had to be turned into cell types, such as blood. They were useful. That's a very inefficient process, as you mentioned earlier, so we decided to sort of explore other avenues of maybe saying, well, if these skin cells can turn into stem cells, why not turn them directly into blood? And so we explored a variety of different ways to do that and found one combination of things that seemed to work quite well.
And how viable is this, what you found?
Well, we've only done it in the laboratory right now. We've done it on multiple samples of human skin, and we focused on the human because we thought there was a clinical application for this. The next steps are going to be clear, and there's a lot of work ahead, of course, to seeing this in the clinic, and that is to do efficient scale-up, larger numbers, not just a dish or a petri dish, but in large numbers of cells, and also to make sure that all the safety and regulation issues are dealt with before we even think about putting this into people.
Tell me about the implications. If your testing goes well, largely well, and you're able to proceed, what kind of implications could this have for people who, well, just tell me what it could do.
Well, McMaster sees that there's a lot of places one could go. We're targeting a particular type of patient, specifically in adult leukemia. That's where their blood system has a genetic mutation that caused them to have a blood cancer. And then getting another source of healthy cells proves to be very difficult. So we thought if we could take their own skin cells, make healthy blood cells, because they do not have the genetic mutation, that they could be used to be transplanted back in without fear of rejection and hopefully replace the bad, quote-unquote, leukemic cells.
Let's talk about how much skin we're talking about and how much blood do you get out of it.
Yeah, it's, we're trying to do, right now we've done this only in a laboratory, but just based on preliminary calculations, and we're very lucky that surgeons already use skin and take skin for purposes of burn. They can take skin from one area of the body, grow it in a dish, and then put it on areas that are infected by either heat or acid burns. So there's some knowledge pre-existing there. We're hoping that about a 4 by 3 centimeters a patch of skin could be removed from a patient, be converted through this process, which we clearly have to optimize, and hopefully have enough to transplant a full-grown adult.
You mean, so in other words, you're getting more blood. It's not like a 1-to-1 ratio of the skin cells you take into blood cells. You're able to reproduce, or you're able to create more blood cells than skin cells?
Yeah, that seems like the calculations seem to indicate that because you remove skin, but the skin cells, once put into a petri dish, before we even perform a conversion process, can be grown and increased in numbers, more than just the patch taken. So that gives you a higher number there. We go through the conversion process, and then once the cells have turned to blood, there's a lot of expertise around the world which already knows how to increase those blood cells that result in higher numbers.
You can multiply the skin cells, then you can convert it, and then you can multiply the blood cells as well. So you can end up with a lot more blood than you started in terms of skin cells. This is fascinating. Dr. Mick Bhatia, thanks for coming and telling us about this as clearly as you have. I know it's much more complicated than you've told us, but it sounds very hopeful, and we will follow your progress and that of your group very closely. Thanks for being here.
Thanks for your interest.